So we still have things to do this evening. We have the Emeritus Professor of Ibogaine, Dana Beal, now. And then you can grab some, those with dinner coupons, grab some dinner. And then we're going to show an amazing film about Gabon, because this all emanates from Gabon, after all. If you haven't seen Gabon Last Dance, it's a really powerful film about the culture of Gabon, and it delves into some Bwiti towards the end. So, Dana Beal. Just to say personally, I've known Dana about three or so years. Huh? Feels like longer. So, okay, so you know your mate's and in with Dana when he starts calling you 20 times a day. He is the most persistent, tenacious bugger I have ever met. You know when you call someone and they don't answer, you might sort of like diminish your interest. It's just 100% all the time with Dana. In fact, it goes up if you don't answer the phone. So, but basically, he funded the Ibogaine movement for 20 or so years. So let's just clap for that. So dinner is sort of ready, but anyway, we'll leave it, and you know, if it's a slightly cooler than we would have expected, that's fine. So anyway, Dana's raring to go. I'll hand over to him anyway. It's going to come up. It's going to come up.
Oh, okay. Anyway, I basically tried to come up with an Ibogaine 101. And I think that all of you should learn at least this amount about Ibogaine and be able to do this PowerPoint. This PowerPoint is actually about 4 drugs, because Wellbutrin is in there as a comparison. But it's also about noribogaine and 18-MC, because it was developed to talk to people who were interested in treating methamphetamine. And we've talked a lot about opiates and talked some stuff about alcohol and cocaine. We haven't really talked about methamphetamine yet. There's a president in the Philippines who's having people shot for doing methamphetamine. In the world right now, there are twice as many methamphetamine users as there are opiate users. The position of the harm reduction movement is that methamphetamine should be accepted. It should be normalized. This is because these guys are really into needle exchange, and methamphetamine is something that is put into needles and shot. But when Carl Hart went to the Philippines and said there's, in effect, no such thing as methamphetamine addiction, he got death threats. And my position is it's a treatable disorder. We have medication that works for it. And you certainly shouldn't shoot people because you won't make the medication available. But there is a real underlying problem, and I get into that here. So anyway, do the first slide. See, this was an article that came out, and the main topic of the article was 18-MC. Next slide. This is the Cochrane Reviews, and all the drugs of abuse, all the treatments for all the drugs of abuse get into the Cochrane Reviews. I don't know whether it's still 56. I think it's more now. But there were 56 studies on methamphetamine, and basically they have nothing for methamphetamine, except in New York now they have supervised methamphetamine use, which is, you know, like supervised injection site. But I'm having a big problem trying to get these guys to even try 18-MC because they can give off-label stuff, but they have to go to the FDA to get permission to give 18-MC. And they are dragging their feet. So anyway, we've already seen this. This is some of the stuff you know already because it was in the previous stuff. But methoxychloronaridine, nor-ibogaine, and ibogaine are the 3 drugs that are out there that are either being sold or being backed by a corporate structure, or they're intending to become a regular approved medication in the United States, and then they will be sold. Next. See, this is an early slide, but you see, here's your ibogaine. Serotonin, you know, serotonin was first discovered, was in the cow gut. Remember Ken was talking about plants? Serotonin makes your alimentary canal peristalse. In other words, you have like a wave that goes down your alimentary canal and moves the food. And that's powered by serotonin. So it's a really ubiquitous thing. And most of your serotonin is actually in your gut. Harmaline, you just complete this little thing here, right? And you get this 3 structure instead of a 2 structure. And it's being produced by the pineal gland as tetrabeta-carboline and phenylalanine, and you're excreting little bits or secreting little bits at night when you dream. So you just change this little thing, and you get something that causes you to visualize. And Naranjo gave this a lot. He actually liked it better than harmaline, or better than ibogaine. And he had to give a little bit of speed with it so people wouldn't just fall asleep and not remember the visions. But it's in ancient soma. There are different varieties of soma. Some soma was based on cannabis, but a lot of soma was based on Pegamon harmala, which is the original little plant. Not Banisteriopsis chiapi, the big plant from the Amazon. It all says the same thing in it. It's in a lot of stuff. Ibogaine's a little bit more restricted in where it's available. But you see, what happens is this little thing here is the side chain that keeps you awake. And you only have to take, instead of taking this and this as ayahuasca, you take this, and it's all in one. Next. Now, he showed, this is a very interesting slide, because he was talking primarily about the ventral tegmental axon here, which basically handles all kinds of impulse and like very rapid response actively, you know, for fight, flight, or fight, and also for food, sex, and water. And this here is implicated in Parkinson's, but it's for vision, because you have to be able to see and move your eyes as fast as you move, or you're going to bump into something. And people who take Ibogaine sometimes get a big improvement of their vision for a while. It doesn't last, but they suddenly can see without glasses and stuff. So this is more affecting vision. Next slide. This is what Ken showed. I can't emphasize this enough. Dopamine is not about directly pleasure. Dopamine is about arousal. Of course, as anybody who's ever had sex can tell you, it's not very pleasurable if you're not aroused. And actually, if it happens to a woman, they often call it rape, right? So, you know, arousal is really important. And next slide. I'm not going to show the third slide, but I want to give you like a a thought experiment. You're out on a windy afternoon. You're in a street in Vienna, and some euro bills come flying by because windy, and all of a sudden money is coming by. What do? You don't even think. You grab the money. You don't want it to get lost. It might belong to somebody. Then you look around and you say, there's nobody attached to this money. I guess I'll put it in my pocket. And of course, the thing is, what is money? It's this piece of paper. You can't eat it. You can't drink it. You can't fuck it. But it will get you all those things. And this, in effect, the little light here, or the bell, right, that caused the rat to know that he was going to get a reward here, so he started alerting over here and early. I'm about to get a squirt of juice on my tongue. That's money to us. Next slide. Okay, now this, we're going to be talking about GDNF a little bit. And this is called the tyrosine cascade. And this is where the dopamine comes from. And it's synthesized into dopa, and then dopamine. And then down here, it's really interesting. It turns into norepinephrine. What is norepinephrine? Adrenaline. So here, food and sex and all fun. Here, fight or flight. But it's all supported by GDNF. Next slide. And this is what I mean. See, here's the part we were talking about before in the brain. But what's this stuff here? That's part of your brain. That's part of your central nervous system. HPA axis. And HA stands for adrenaline. Mice that did not have the gene for GDNF because it was knocked out as an experiment. It's called knockout mice. Were born with no kidneys. They were born with no kidneys. So in terms of the GDNF is probably like, involved in a lot of peripheral stuff. But the kidney is like a scaffolding for this little piece of the brain that directly affects the heart. Right? When you think about cardio problems, why would cardio problems come up? Well, it's because it's directly connected to the adrenaline, which is directly connected to the brain. And, if you need, you've heard the expression scared to death. This literally happens sometimes. People are scared so much that they have a heart attack and die. If you need to get away from something in a hurry, let me tell you, that jolt of adrenaline can be really useful. Next slide. Okay, here is sex. And this it kind of explains what happens with arousal. Here's where the sex starts. This is a male rat, so he stuck it in. And he does it for a little while, and then he starts feeling really good. So then he gets aroused, and he goes up to a whole new level of pleasure, right? This is the really good part, right? And then he has an orgasm, and it rapidly goes down. They have a cigarette and they fall asleep. Next slide. But see what happens when you have something, and this is the thing about amphetamine and cocaine, right? They're directly affecting the switch. They're directly affecting that synapse between incoming dopamine neuron and outgoing dopamine output. And what's happening is the cocaine, or Ritalin, is blocking reuptake, whereas amphetamine is really messy. It just causes you to squirt big amounts of dopamine for 12 hours. And what happens when you flood this thing? It's roughly analogous to what happens when you walk out of a really darkened room into really bright sunlight. You have to almost grab your eyes. Your eyes start watering. You can't see, right? But after about 3 minutes, you get used to it. And if you go back into the place which is dark, you can trip over the table that you're supposed to know is there. And what happens is there are some people, this is the distribution of people who become addicted, where this whole thing is kind of blinded and they don't really recover. They built up a tolerance, and the only way they can get a dopamine level enough to be aroused, enough to get it together, to get up in the morning, right, is to take stimulants. Next slide. And this is what happens. This is just like 100 milligrams of meth. And you see up here, there's just more of these dendrites here. And here, there's less of these cell bodies and just less of a tracery of dendrites. So you're losing some connections there every time you take meth. And, you know, people shoot meth, so pretty dangerous. Next slide. Okay, now, this is the thing that got me in a lot of trouble at first, because this is back when we did the Ibogaine book, the Ibogaine story, of which I only brought one copy. I should have brought a bunch of copies. Nobody will be able to buy it. But it's from a thing called quantum coherence in microtubules. And this is supposed to be a microtubule next to a nucleus. And microtubules are fiber optical conduits in the brain. They're little, you know, like 25 nanometers wide tubes. So you have all these fiber optical conduits, and they, due to something called superradiance, convert all kinds of body temperature, Brownian motion, any form of energy into this very faint but laser-like, in the sense that it's all, you know how lasers are, you know, serried ranks of light instead of like light bouncing all over the place. You get coherent light. I thought, if you have something that's like based on photonic activity, and we were talking about from the French at the very beginning, 1991, we were talking about a neurophrenic or a neric or remogenic or REM-mimetic brain phenomena. I was saying, if we have photonic activity that is coherent, that's holding the whole brain together, so you're not thinking billions of little thoughts, you're thinking only one thought. It's like kind of rich, and it's got a lot of different parts. But you have one coherent after another, and it's flickering, right? It's what's called emergent. This would be a perfect basis for it to be occurring. Next slide. And these are actually records of different coherent states, of different moments in calcium. And it's distributed throughout the brain. So your brain is taking photographs of the coherent whole, one right after another, in every situation you're in. You're storing that away. Now, I had a fatal heart attack, and I was frozen after they revived me, because I was dead too long. I was frozen for 6 days, and I woke up and I had absolutely no memory of the day that it happened. I don't remember going to heaven or any of that stuff. If you're frozen, the calcium chemistry at 86 degrees cannot form permanent memories. But you see, the other problem is, it's like photographs. It doesn't change. You have to change your interpretation of what happened, right? It's still the same thing happened. But, you know, for instance, you decide that the person who was my enemy is now my friend. oh, this guy that I thought stole from me, it was really this girl over here that stole from me. And you go back and you reinterpret everything that happened. But the basic visual memories continue unimpaired to the end of your life. Next slide. And so about the time that I published the book, there was this woman named Nancy Wolfe who did a follow-up on quantum coherence of microtubules, which is where that came from. And basically she's saying this stuff has to be broken down, and you have these neurotropins that are causing the regrowth of, see, calcium. This is all calcium stuff. Stuff is being broken down and regrown. And the agent of it is neurotropin. So see, we had, when we were dealing with NIDA, and they really didn't want to believe this could work, we already had the knowledge that we needed, but it was not called to our attention in time to persuade the National Institute on Drug Abuse that they should continue the program. Next slide. So anyway, in 2000, there were a bunch of explanations or potential mechanisms of action of Ibogaine. There were simple agonist-antagonist actions. An agonist-antagonist you can think of as turning off a light or turning a light back on. It lasts for a certain amount of time, and then it's gone. Neurotropins, which we just barely kind of knew in 1993 that there was such a thing as neurotropins, and it gradually only became clear, and they were, let me see, they were first looking at like GDNF for Parkinson's, and they couldn't figure out how to inject it into the base of the brain. And then they were looking at it for alcohol. Maybe this is why Ibogaine works against alcohol. And there was this deficit that we couldn't really understand. Now this was the first thing was actually discovered, is the kappa opioid effect. And this is actually a different peptide. And it is not like mu. It's not like the thing like, heroin. You have dysphoria and protection from central nervous system depression, which can actually kill you through an overdose. And you have, see, this is perfectly understood if you understand how you have an accident, and first you have excruciating pain, and you get a big surge of mu, right? Because your body is making plenty of endorphins. You could actually die from respiratory collapse. So here comes the kappa opiate to stop the respiratory collapse. But then what happens next? After a couple days, your endorphins wear off, and you're in a lot of pain. It flips around, and it augments the effect of mu opiate. And this is the really interesting thing that we didn't find out until relatively recently, and this came courtesy of the noribogaine people. It downregulates something called beta-arrestin. And beta-arrestin is from the guanine-coupled protein as opposed to the ATP, ADP. What happens is, this is a bunch of different systems, not just opiates. It's something like dopamine neuron. What happens is that you overstimulate a particular system, and you can't keep the organism at a state of continuous high arousal. Homeostasis won't allow it. So the receptor protectively secretes something called beta-arrestin, which means that now only every other impulse triggers the receptor. Well, this, if you're like patient D, you need your Cinemet to work, and your Cinemet is stopping to working. In the future, what they're going to do is they're going to give a tiny bit of Ibogaine with all Cinemet because it gets rid of tolerance. It abolishes or reverses beta-arrestin. Next slide. This is a pretty important thing, and this is like important because Stanley Glick, who invented AT&MC, says this is the main effect of Ibogaine. And that is it blocks the nicotinic acetylcholine receptor so that you can, I guess, have plenty of muscarinic acetylcholine, which enables you to visualize and dream. And you have to turn off a bunch of like waking systems like dopamine and serotonin and nicotinic acetylcholine in order to sleep. And all the stuff that is happening is cholinergic, but it's not through the waking part of the cholinergic, it's through the sleeping part. And see, this here is the most, is a really, really common anti-smoking drug, but it's basically alpha 3. No, this is not. This is a mistake. It's alpha. Let me see, what is it? Alpha 4 beta 2. Sorry, I just made this slide. And the other thing it does, it's got this habenulus, which I'll get to in a minute when I get to the AT&MC slide. It modulates the receptor through, it modulates the dopamine system, through a parallel structure that is right next to it. I'll show that to you in a minute. Next slide. And here's the 5-HT2A, which gets everybody in trouble. Because if you have this in your drug, they're likely to ban it as a psychedelic drug. And it seems to be actually to really correlate with the expression of it really correlates with the expression of GDNF and BDNF. You have SSRIs that express GDNF, but they don't cure addiction, and they're not patenting them as a cure for Parkinson's. So this turns out, if there's an anti-addictive effect, which Beckley claims for psilocybin, it's 5-HT2A. But AT&MC was designed not to have it. Next slide. Okay, now this is something that appeared in, and this is a possible explanation of how noribogaine could be working, or ibogaine could be working without having growth factors. And the acetylcholine actually, it says it inhibits the activity of dopamine neurons. That's not exactly true. What it does is it inhibits like regular waking activity. And if you got like muscarinic acetylcholine, you're functionally in a state like an ibogaine trip where you can't move very much. It's like when you're asleep on the couch, but you're not really asleep on the couch and you're going in and out of like a dream. Like 1 minute you're dreaming and then you kind of wake up and you're aware of stuff that's in the room, then you go back into the dream. The serotonin, it has to be, if you have serotonin, you can't have muscarinic acetylcholine. So it isn't slowing the activity of dopamine-making neurons at all. What's happening is the dopamine, see, this is addictive drugs stimulate dopamine-producing neurons in the substantia nigra and the ventral tegmental area. Nor-Ibogaine, which binds to the receptors in these neurons, inhibits dopamine release and limits its effect on other areas, including the habenula. Well, it's not inhibiting dopamine release at all. What's happening is that you're switched over to a sleep state, a kind of a sleep state, and in sleep state, dopamine is naturally inhibited, because if you had dopamine, you'd be awake. And so what's actually happening is that your dopamine function is being repaired, and when you wake up, sometimes after just a few hours of sleep, you feel pretty good, and you don't feel the feeling of I can't get anything together that people have with unassisted heroin withdrawal. Next slide. Okay, so this was the thing that seemed great that we were getting rid of this. This is what makes you sick on Ibogaine. It gives you, it makes your inner ear acutely sensitive, so that if you try to move, you get car sickness. And it also is implicated in tardive dyskinesia, which, if you take too many antipsychotic, stuff like Haldol or Thorazine, you end up with this rocking motion that you'll have for the rest of your life. It's caused permanent damage. And it's interesting, Ken says this is another source of QT prolong. But it also blocks NMDA toxicity. And there's a guy named Olney in 1998 patented Ibogaine as an anti-stroke medication. And this is before we knew that GDNF can really fix the brain up. Next slide. Now this is very important, and it's been discounted, but I think it's one of the actually key effects of Ibogaine. And this is the same effect as ketamine. And ketamine is being used to treat addiction. People are injecting ketamine, people are taking ketamine. And that is because it cuts both opiate withdrawal and super-sensitization to stimulants. Super-sensitization is the thing where you eat one peanut or one potato chip and you can't stop. Well, you have one line of cocaine or one toke of meth and you can't stop, right? So in London, last time I really did anything with Hattie Wells, and I think early 2002, the little Ibogaine thing we had, I speculated, I said that we know that REM mimesis makes the brain plastic and makes it grow back. Perhaps this mechanism where you can get some trickle of calcium in is enabling the cell to get some calcium to rebuild some dendrites. Next slide. And there you have it. See, this is what I was visualizing, but I didn't have the slide done. But you see, this is all calcium. And you need some calcium either from, stores within, recycling other dendrites, or you need some from outside. Next. So anyway, what do we have with GDNF? This is definitely the most powerful effect of Ibogaine. Think about it. It takes 300 milligrams of Wellbutrin to have a dose of an anti-smoking medication. It takes only 4 milligrams twice a day to have the effect we saw in patient D. So this is a really strong effect, 12-fold within 6 hours. And I don't think that any of the other effects, like we just saw, are really going to be triggered by 4 milligrams. You have, I think, just a residual, small amount of stimulation at 4 milligrams. But the GDNF is happening. So if you want to know, what is the signature effect of Ibogaine? It's right here. 48 hours. They weren't talking, and now they're hooked up like the internet. Next slide. See, now this is a slide I put back in because this shows how it works ordinarily. Ordinarily, you have a GDNF that comes along, right, and marries this receptor, and it activates something called an RET, which is a subsurface receptor. And this actually goes through about 20 stages down to Krebs. These are extracellularly regulated ERKs, extracellularly regulated kinases, and then at the bottom of, when you get right down to the cell nucleus, you have this thing like a skyhook that comes in and unfurls the DNA the way Roman showed earlier, and a bunch of like messenger RNA comes out and is hanging out in the intracellular space. And then, see, this is ordinary. This is ordinary. This is like something like serotonin. The actual, this is dopamine. The dopamine is actually manufactured right here near the surface of the cell. You get tyrosine and stuff coming down here, and there's a cascade, but it's manufactured right here. Peptides, including very big peptides like neurotrophic factors, you have to actually express messenger RNA from the cell nucleus, and then you have a pre-propeptide, and then that causes a propeptide, and then it's stored for a while and worked on in these trans-Golgi networks, and it ends up in large core vesicles that are like right near, the core of the main core, and it's extruded into extracellular space and can actually be carried by blood to a distant location. Because remember, endorphins are peptides, smaller peptides, but you can generate endorphins and it'll go to the place where it hurts and it will stop it hurting. And likewise, GDNF, next slide, goes all over the place. Now here, it shows, once again, this 12-fold increase. It's really big. Next. And here is the question. How does Ibogaine do it? They didn't know. They just didn't know how it was doing it. Now, what we found out more recently is Ibogaine is unlike a lot of other compounds, that it gets through the cell membrane and actually works on a subsurface receptor right here and turns it on directly, so that you don't need the GDNF. And what you're making is you get a lot of these ERKs, right? And they come down and they're just, a huge flood of GDNF comes out. And it just floods all the cells with GDNF. Next. So anyway, they were cruising along, and I thought, oh, ATMC is, also making some GDNF. It seemed like an explanation. But then we had this paper that occurred in 2010, right when a guy was writing up the big article for the Village Voice, which says Ibogaine, like Ibogaine, expresses GDNF, but 18-MC does not. Wow. Now it's really beginning to look like maybe the psychedelic effect is paired with the growth factor, and at the same time, it turns out that 18-MC doesn't have QT prolong. Well, is the QT prolong kind of the cost of doing business to make GDNF? Next slide. So here's the answer. This was Ken's abstract, and what they found was that Ibogaine and noribogaine and voacangine all block something called the human ether agogo related gene, and that they call it ether agogo because fruit flies have this gene, and the presence or absence of it makes them act like they're drunk. So they called it ether agogo because they were giving the fruit flies ether. And 18-MC is possibly a safer conjugate and a candidate for development. The ibogaine and the noribogaine have levels associated with QT prolongation and fatalities. And this is something that Ken came up with because we noticed that the witching hour in treatments, when you really got to watch them, is not in the first 12 hours. The deaths were all occurring between 14 and 22 hours, which is when the ibogaine is gone, but they have plenty of noribogaine on board. And noribogaine is kind of clunky and hard to clean out of the system. Next slide. So, 18-methoxychloronaridine, all right? It was, now what this means is it was developed by a process of political synthesis. In other words, we don't want to use a psychedelic drug, so we're going to make a non-psychedelic drug. That was the rational development. And it differs from ibogaine for 3 positions, and this is the main thing here. This is the thing that kind of, this is a carboxyl. This kind of like just neuters this wonderful molecule, so it doesn't do a whole lot of things. Next slide. Now, it does do some things. Here's a rat that's been given, well, different rats were given methamphetamine and nicotine. And this is like a 60% reduction in methamphetamine. Remember I said nothing worked? The Wellbutrin seemed to work a little bit, but everything else they tried, mantidine, things like that, they did more crystal meth. And this is amazing. Look at this. This'll knock Wellbutrin out of the box because it's working directly in the part of the brain where the addiction occurs, and Wellbutrin works all through the body. Next slide. And so basically, this is a pretty big, complex thing. And Glick said, the only important thing is right here. It's called the habenula, and everything is explained through the single effect of alpha-3 beta-4 nicotinic acetylcholine blockade. This is a pretty complex place. You mean you found this one little place that can do all this stuff? This is like flakier than MASH's theory. Next. But see, here's your Wellbutrin. See, this is not like a 40% effect. This is a 10% effect. So, you know, you barely can tell that it had an effect, but they're selling it. And I was in jail. Guys were getting Wellbutrin and crushing it up and snorting it because they couldn't get cocaine. Next slide. Here's a different way of saying the same thing. All right, but it rebounds. Notice it came down for methamphetamine, but then it rebounded. Next slide. It came way down for nicotine, but then it rebounded. Next slide. This is what Glick said. He said the hallucinations are just an unfortunate side effect, explaining that Ibogaine works on the brain like a hybrid of PCP, that's the NMDA, and LSD, that's that 5-HT2A receptor, that naughty receptor that gets you high when you take magic mushrooms. Part of the problem is that when you go through this thing, it's so profound, you got to believe it's doing something. Well, actually, maybe I changed my mind about doing drugs. In part, it's an attempt by the person who is undergoing it to make sense of the whole thing. Generally speaking, Glick's research on rats has shown that Ibogaine dampens the brain's so-called reward pathway. Well, no, this is, trying to fit a brain that contains 30 different neurotrophins into a paradigm where you only have agonist, antagonist, simple stuff, effects that wear off after a day. And he's calling it, you see, it dampens the brain's reward pathway, reducing the release of neurotransmitters like dopamine. That's not what happens. It's basically making you more sensitive to the dopamine you have, which causes the highs associated with everything to heroin to sugary foods. And see, so they're back to the original Nancy Reagan idea that dopamine is the pleasure switch. Well, we already saw it's definitely the money switch. Some people probably, maybe Trump is like McScrooge, and he goes into a room somewhere and rolls in his money. The compound has been proven to increase production of GDNF, a type of protein that quells cravings. This is completely misleading. And to block the brain's nicotinic receptors, the same spots that are stimulated by tobacco and other addictive substances. This is nonsense. Next slide. This is what's making them upset. See, this is from the movie that Ben DeLonin did, Rite of Passage. And the guy's on a bed, and he's got shades on so that the light doesn't bother him. And he's completely a threat to no one. Why do they have to make this illegal? Next slide. Because of this. This is awake, right? And this is REM sleep. They're actually pretty similar. This is a little bit coarser grain, but you can see all these other things. Now, the interesting thing is that all of these effects are quantum coherent effects. This is a complete global state of the brain that is, once again, coherent, even though it's shut off so that your brain can function like a quantum computer and do all kinds of repair work that happens while you're asleep. But without REM, you can, I'll give you an example. They had rats, and they jolted them with electricity every time they started to go into REM. And the rat's immune system completely collapsed, and they died due to the bacteria that was in their stomach. So I don't know why there's so intent on shutting it down, because it's a really beneficial state. Next slide. And I've explained this a bunch of times, but I got a new way of explaining it. This shows the side of a dendrite, and acetylcholine. Acetylcholine is this really interesting stuff. It's manufactured at your mitochondria. Mitochondria is a little energy factory. Acetylcholine, the mitochondria takes choline, which is sucked back into the cell, and attaches an acetyl group, which is like vinegar. And what happens is, you can see this is very close. This acetylcholine is right on the surface of the microtubules. The glutamate heats stuff up. Glutamate is like kind of kept away from it by the spine because you don't want to singe. This is basically a conduit full of jelly, and there's waves occurring in it. How do you make a wave into a particle? You have to observe it. You have to precipitate it as a particle. So what you do is you flash some infrared from glutamate, and when you have like a drug relapse, okay, let me go longer. When you have a drug relapse, when you have a drug relapse, you have a flare of glutamate that causes you to visualize. The visualization actually occurs through the acetylcholine, and it gets so strong that you have to go do drugs. Next slide. Okay, I'm going to do it really fast. Okay, what this shows is that the most primitive one-celled little protozoa that's moving around just with cilia, despite not having a central nervous system, somehow is able to do that thing with quantum coherence of light, model its surroundings, encounters a stone, backs up, and goes around it. Next slide. And this is the really interesting thing. See, I got this new stuff, man. Give me a chance. I got new stuff here. This shows 3 kinds of ways that the GDNF can work. It can be released by a neighboring cell and work on nearby neurons in the absence of formal synaptic connection. A neurotrophic factor can act on the neurons that release it, or it can be released from the terminals of a nerve cell that acts on synaptic terminals of these terminals. So you're making me go too fast. The mode of transmission that predominates in the adult and spinal cord is yet to be determined. Next slide. Okay, so anyway, this is the PGO wave. And the interesting thing is, this is what starts REM, and PGO waves have been detected when people are tripping on Ibogaine, breaking into regular waking consciousness. So they're actually, the people who are saying that it's not REM, that's just like hallucinations, whatever that means, they're not actually scientifically correct. And this comes from the pons. This is where, stuff is transmitted from the eye back to the occiput, where you actually kind of register it and process it. And these are all PGO waves. And you have like PGO-like activity on Ibogaine. Next slide. This is the other really important thing that's left out of the way that, like the habenula or the MASH explanation, which is that in addiction, what happens is you go from the prefrontal cortex controlling the cerebellum, which is the back part of the brain where you learn how to ride a bike, so that you can ride a bike down the street and talk to somebody and not have to pay attention to like riding the bike. Your brain just does it for you. What happens is that the cues for drugs turn on the cerebellum directly. And there's a stuff that we found when we were looking at the neurotoxicity, which actually doesn't exist, but it was showing where the activity is. Neurotoxicity, you have to take 200 milligrams per kilogram or something. That doesn't ever really happen. But somehow you're getting a, in the process of seeing everything and, reevaluating everything, these cues are actually being, it's like you shined a light on them and returned them to the control of the prefrontal cortex. Next slide. Okay, so this stuff here, this is the future, because of GDNF. This is Dalibor Sames. This is the same stuff that What's-His-Face was talking about. Ignacio Carrera. Next slide. And here's how they did it. They like took this little thing out. They have a whole way of like cutting out a piece of internal lattice, which never would have occurred to me. And this is the final slide here. See, it didn't take that long. The most powerful compound is also the most toxic. So there's some kind of relationship between toxicity and the amount of GDNF that's being like generated, and if you try to do it too fast or go too high, it won't necessarily cause hERG blockade, but it really has to be studied if any of these are cytotoxic. And one of the things I told Ignacio was you gotta look not just at cytotoxicity, you gotta look at cardiotoxicity. So anyway, what I want you all to do is to make your own Ibogaine PowerPoint and be prepared to give this presentation to the media, to politicians, to scientists. Don't be intimidated. Learn the science. I'm only a reporter. I worked on this story for a long time, but I have no PhD. Anybody can do this, and we have to, you have to be a barefoot doctor. You have to get the medicinal facts out there. This is not working because it's blocking some receptor somewhere. It's working because it's causing the brain cells to grow back. Thank you very much.
Don't you want to have his energy when you're 70? Okay, we've got time for a couple of quick questions. I know you're all starving and it's taken a long time this evening. A couple of quick questions. Lily at the back. One question from Lily and that's it.
Well, it's because you have to be able to remember. And they have to be similar enough that memory, that visualizations can be passed from the sleeping state to the awake state. You remember your dreams. And if you didn't remember your dreams, it wouldn't really re-edit your personality, your waking consciousness, all the stuff we do dreams to do. Yeah, well, I mean, all dreams to some extent are lucid because you have some memory of what you dreamed at night. And it's really important to you. It doesn't make any sense to anybody else. But this is the brain working as a quantum computer. Now, I did want to talk about one thing, which is, which is the immune system. The immune system apparently is modulated by REM. And I just want to put the idea out there that somehow the brain, which orchestrates when you're a fetus, that thing where it grows your kidney is being orchestrated by your central nervous system. Your brain is in continuous fetal REM. And we might have a way to study this and figure out how the brain is telling immune cells, hey, you're attacking healthy tissue. You're supposed to attack the cancerous tissue. So, we've seen a lot of things like the effect on multiple sclerosis and the effect on Crohn's that Ibogaine sometimes has. And I don't think it's happening just because of the GDNF. I just have a sense that it's the thing that's driving the GDNF, that's the thing that causes the GDNF to be expressed in a tenfold increase. Okay, thank you.
Great. Thanks very much, Dana.